All right, let's dive into a fascinating world today, shall we? We're talking about nematodes, these seemingly simple little worms, the kind that wriggle around in the soil. But don't let that fool you. They're actually incredibly successful. I mean, they've managed to colonize pretty much every corner of the planet. And speaking of successful show, one of our listeners sent in some research comparing two specific species. And
Yeah.
this research really highlights just how much there is to learn, even from these tiny creatures. You've probably heard of Caenorhabditis elegans, or C. elegans for short.
Elegans is a superstar in the world of biology. It's this model organism that we've learned so much from.
Kind of a lab rat of the worm world, I guess you could say.
The development is like clockwork.
Mm.
I mean, we can track every single cell as it grows from an egg to an adult.
Yeah.
So in that sense, it does seem pretty straightforward. But
Right.
as you mentioned, they're incredibly adaptable. They're found everywhere. They've clearly figured out how to thrive in a wide range of environments.
That's what makes this research so intriguing. You see, this study compares C. elegans to another nematode called Pristionchus pacificus. Now, both of these species belong to a group called Rhabditina, so they're actually fairly close relatives, evolutionarily speaking.
Kind of like cousins, yeah.
Exactly, but like cousins who grew up in totally different neighborhoods, they've developed some really interesting differences.
And I think that's what highlights one of the most exciting things about evolution is that even closely related species can take these different paths and come up with unique solutions to survive and thrive. It's really remarkable.
And one of the most obvious differences between these two nematode cousins is in their mouths.
Okay.
Believe it or not, C. elegans has this structure in its pharynx called a grinder.
I see.
Basically, imagine like a tiny little food processor built right into its throat, and this grinder, it physically breaks down the bacteria that C. elegans eats.
Fascinating. So it's like pre-digesting its food in a way?
Exactly, and that's why if you look at the gut of C. elegans, you don't usually find a lot of intact bacteria just hanging out. They've been pretty much pulverized by this grinder.
Okay, so now the question is, what about P. pacificus? It doesn't have this grinder. It lost it at some point during its evolution.
Right.
So how on earth does it break down bacteria and get those nutrients?
Yeah.
It's a completely different approach to feeding. And that's what really makes this comparison so fascinating.
So the researchers in this study decided to tackle this question head-on by looking at the transcriptoma of the intestine in both species.
Okay.
And for our listeners who might not be familiar with the term transcriptomo, it's like taking a snapshot of all the RNA molecules that are active in a particular tissue.
Right, like a molecular inventory.
Exactly. And RNA is like the messenger molecule. It carries instructions from DNA to build proteins. So by looking at the transcriptome, you can see which genes are switched on and working hard in a specific part of the body.
It's like looking at a factory and see which machines are running and what they're producing.
Perfect analogy. So the goal of this research was to figure out what these differences in gene activity can tell us about how C. elegans and P. pacificus eat, and maybe even how those differences might affect other aspects of their lives, like how they deal with bad microbes.
Right, because if the way they're breaking down their food is so different, you'd expect that to have downstream consequences for their whole physiology.
So to do this, the researchers, they isolated the intestines of both C. elegans and P. pacificus.
That's pretty delicate work.
It is. And then they extracted the RNA and sequenced it, which basically means reading the genetic code to figure out which genes are being expressed.
Like deciphering the blueprints of the intestine.
Exactly. And they also did this for the whole worm to get a baseline measurement so they could pinpoint which genes were specifically more active in the gut compared to the rest of the body.
It's like comparing the workforce of the entire factory to the specialists in a specific department.
So they've got these lists of genes that are really busy in the intestines of both worms. What did they find when they started comparing those lists?
Well, right off the bat, the numbers themselves were pretty interesting. They found 724 genes that were significantly more active in the intestine of C. elegans compared to the rest of its body.
Okay.
And for P. pacificus, that number was 464.
So already a bit of a difference there.
Right. And importantly for C. elegans, the data largely matched what had been found in previous studies. About 66% of the genes they identified had already been recognized as being highly active in the intestine. So it's good to see that consistency.
It gives you confidence in the results.
Absolutely.
Mm.
But here's where things get really interesting. When they started looking for overlap between the intestine-enriched genes of the two species and specifically focusing on one-to-one ortholics,
And just to remind everyone, orthologs are genes in different species that evolved from a common ancestral gene.
Right, so you'd expect some overlap there, but the amount of overlap was incredibly small.
Like shockingly small.
Yeah, out of thousands of orthologous gene pairs, they only found 15 that showed a significant increase in activity in the intestines of both C. elegans and P. pacificus. 15. And statistically, this was highly significant. The p-value was way below any reasonable threshold.
So it wasn't just random chance.
No, not at all. This suggests that even though both species have an intestine, the way they're using it at a genetic level is vastly different.
So they're both doing intestine things, but they're using different sets of genes to do it.
Exactly. And that points to a huge functional divergence in their intestines. Even though the fundamental role of the intestine is the same to digest food and absorb nutrients, the specific genes, and therefore the molecular machinery carrying out those tasks, seem to have evolved in very different directions.
Okay, so a lot of differences, but were there any common threads at all? Besides those 15 genes, I mean.
Well, yes, and this is fascinating. Among those 15 conserved genes, 3 of them are involved in what's called hedgehog signaling.
Hedgehog signaling.
It might sound kind of strange, but it's actually this incredibly important pathway for development and tissue function in a huge range of animals, including humans and insects. Yes, and in the context of the intestine, it plays a key role in how the intestine develops and how its cells communicate with each other.
So even with all the differences, this pathway seems to be a common thread.
Right? It's like this ancient molecular conversation that's essential for building and maintaining a healthy gut across so many different species.
That's amazing.
And the researchers even highlighted a study in C. elegans where disrupting the PDR genes, which are part of this pathway, actually led to abnormalities in the intestine. So it really reinforces the idea that hedgehog signaling is crucial for intestinal health, even in these 2 nematode species, with their vastly different lifestyles and feeding mechanisms.
So hedgehog signaling is a common thread, but what about zooming out a bit? What about looking at entire gene families instead of individual genes? Did they find more overlap there?
You'd think so, right, but actually the general trend of divergence continued. They look at which gene families showed the highest overall expression in the intestines of each species.
So basically adding up the activity of all the genes within a family to see which families are the busiest.
Exactly. And while some families, like cysteine proteases and aspartate proteases, which are involved in breaking down proteins, were highly expressed in both species, many others were specific to one or the other.
So even the toolboxes they're using to digest their food are largely distinct.
Yeah, it's really striking. And to make things even more granular, they looked at protein domain families. These are like functional units within proteins. You can think of them as Lego bricks that each have a specific function, and they can be combined in different ways to build different proteins.
I like that analogy.
So when they narrowed it down to protein domain families that were significantly enriched in the intestine-specific genes of both species, they only found 5. CUB domains, cytochromes P450, C-type lectins, collagens, and VWA domains.
5. Still a pretty small number considering all the complexity of the intestine.
Right, and those families, they tend to be involved in pretty fundamental processes. For example, in C. elegans, CUB domains are often found in proteins that break down other proteins in the gut.
Okay, so digestion.
Right, and cytochromes P450 are a large group of enzymes that are involved in various chemical reactions, especially oxidation reduction processes, which are important for processing and sometimes detoxifying different substances. C-type lectins are often involved in recognizing carbohydrates, and they can play roles in immunity and cell signaling. Collagens are structural proteins. They're essential for building and maintaining tissues, and VWA domains are found in proteins with diverse functions, often related to cell adhesion or interactions between proteins.
So even the shared families seem to be involved in pretty basic functions that you'd expect any intestine to need.
Yeah, things like breaking down food, dealing with the chemical environment, maintaining its structure. Makes sense.
Now, we talked about that big initial difference, the grinder. Did the researchers look into whether this anatomical difference led to different conditions inside the intestine, like the pH level?
Oh, absolutely they did.
Mm-hmm.
They hypothesized that the different ways these nematodes process bacteria, with C. elegans physically grinding them up and P. pacificus doing something else, might actually create different internal environments.
Right. Different levels of acidity.
Exactly. And to test this, they actually measured the pH gradient along the intestine of P. pacificus
Yeah.
using these tiny pH-sensitive nanosensor.
By nanosensor.
Yeah, super tiny sensors that they injected into the gut. It was like microscopic surgery.
That's incredible.
And the results were surprising. Despite all the differences in gene expression, the pH gradient was remarkably similar in both species.
So it's basically the same.
Pretty much.
Hmm.
They both had a pH of around 6 in the anterior part of the intestine, the front end, and it gradually became more acidic, dropping to around 3.5 in the posterior and the back end.
Wow, so even though P. pacificus doesn't have a grinder, the way it's breaking down bacteria and extracting nutrients seems to work under pretty much the same acidity levels as in C. elegans.
It suggests that the fundamental chemical environment needed for digestion might be more conserved across these species, even though the specific genes and molecules involved are so different.
Okay, so different eating methods, similar gut pH. What about how these nematodes handle those nasty microbes in their surroundings? The study mentioned that the lack of a grinder in P. pacificus could affect how easily they get sick from pathogens. Did they look into that?
They did. They compared the intestine-enriched genes they identified to genes that are known to be involved in the immune response to pathogens. Things like Bacillus thuringiensis and Staphylococcus aureus. So the idea was to see if the genes that are busy doing intestinal things are also the ones that get activated when the worm has to fight off an infection in its gut.
Makes sense. The intestine is the first line of defense against anything you swallow.
Right.
So what did they find when they compared these two sets of genes, the intestine genes and the immune response genes?
The results were pretty clear-cut. In C. elegans, there was a really strong overlap between the two. For 6 out of the 8 pathogens they tested, the genes that were active in the intestine were also heavily involved in fighting off those infections.
So for C. elegans, the intestine is not just about digestion, it's also a key player in immunity.
Exactly. But here's the contrast. In P. pacificus, none of the pathogen comparisons showed that same strong overlap. There were a couple that were borderline significant, but nothing like what they saw in C. elegans.
So in P. pacificus, the intestine doesn't seem to be as directly involved in the initial immune response to these pathogens?
That's a good way to put it. And it actually lines up with what we already know about P. pacificus. It's generally more resistant to some pathogens compared to C. elegans.
Interesting.
So the researchers propose that maybe this lack of a grinder might actually be helping P. pacificus fight off infections.
But wouldn't the grinder be helpful for smashing up bad bacteria?
Well, it's a bit of a double-edged sword. While the grinder does break down bacteria, including the harmful ones, the researchers think that in C. elegans, this sudden physical disruption might also cause a rapid release of bacterial toxins in the gut.
Oh, I see.
And that could overwhelm the worm's defenses. But P. pacificus without the grinder might avoid this sudden onslaught of toxins.
So maybe it's taking a more gradual approach.
Right. However, it's worth noting that the grinder in C. elegans does act as a physical barrier too. It can prevent some pathogens from easily getting a foothold. And there's evidence that if you mess with the grinder's function in C. elegans, it can sometimes make them more susceptible to certain gut infections.
So it seems like there are trade-offs to both approaches.
Exactly. It's a complex interplay between digestion, immunity, and the types of pathogens they encounter.
Makes sense.
The researchers also bring up the fact that both species probably have different ways of managing their gut microbiome, that community bacteria living in their intestines.
Right.
And disrupting that balance, whether it's through a malfunctioning grinder in C. elegans or other factors in P. pacificus, can make them more vulnerable to pathogens. They also pointed out that we still don't fully understand how long bacteria stay in the guts of either species, which could also explain some of these differences.
So lots more to investigate. What was the big takeaway from this study? What did the researchers conclude?
Well, their main point was that this research provides some really valuable insights into how feeding systems evolve and how those changes are reflected in the genes that are active in the intestine. They suggest that the loss of the grinder in P. pacificus might have been a key adaptation that allowed it to explore new ecological niches, like decaying beetle carcasses.
They even have a name for that lifestyle. Necromenic?
Exactly. So ultimately, this study really highlights the incredible diversity that can arise in evolution, even in relatively simple organisms like nematodes.
And for you, our listener, the key takeaway is that even though nematodes might seem simple, they can have surprisingly complex and diverse internal workings, especially when it comes to something as fundamental as digestion and immunity. And the fact that these two species have such different gene activity in their intestines, despite having a similar pH environment and sharing that ancient hedgehog signaling pathway, It really shows you the power of evolution to come up with multiple solutions to the same basic challenges.
It really makes you wonder what other hidden complexities are lurking in the nematode gut, doesn't it?
It does, and that leads us to our final thought for you to ponder. Given that these nematodes have this conserved intestinal pH, but such different genes involved in digestion and immunity, what other undiscovered mechanisms might be at play in there? And could these findings from these tiny worms actually be relevant to our understanding of our own digestive systems and our complex relationship with the trillions of bacteria living in our gut? Something to think about. We hope you enjoyed this deep dive and that has given you some food for thought, pun intended.